we're going to talk about some of the important highlights from Chapter 3. Chapter 3 starts off with Section 1 talking about microscopes, and I feel like that's pretty cut and dry, and you can look at that yourself. And it ends with this section. I need to go back to it. It ends with the cell theory. And cell theory is so interesting how it came about. There were so many cool people that worked on this to discover cells, but essentially this sentence here is pretty important that all living things are made of cells, cells are the basic unit of life, and all cells arrive from other cells. So that is cell theory. That is known as cell theory, and it's really important that everything that's alive that we know of is made of cells, and that all cells come from cells, and that's the smallest form of life that we have classified. So moving forward from there, there are two main types of cells that we are going to talk about. One is the prokaryotic cell, and prokaryotic cells are generally just bacteria and archaea that have this very basic cell type. And this is just one example that's right here, but there's lots of other examples. So prokaryotes are very basic, and they were the first cells that we know of here on Earth. They were here for billions of years before eukaryotes were. Prokaryotes are single-celled, and they do not have a nucleus. Their DNA right here is just kind of kicking around inside of their body. It's not encased. Because of that, and because there's no protection on its DNA, because of that, bacteria can actually be mutated a lot easier than eukaryotes because it's unprotected. So when it's around heat or chemicals or radiation, it can mutate a lot easier because it just doesn't have that extra protection of being inside of what's called a nucleus, and we'll talk about that in eukaryotes. However, it also is kind of complex internally as far as its metabolics. The metabolics inside of here, like how the chemistry is happening and fluid exchange and different things moving throughout it, gets pretty complicated because everything is floating around together. And so if a reaction has to happen in one area, it's going to kind of spread to the other area as well. So because there's no compartments inside of it, prokaryotes are very complex metabolically. Okay, so I don't know if I said this, but prokaryote, this word prokaryote, that actually means before a nucleus. Okay. The next thing that I want to talk to you about is size and scale. They have this in your book, but actually University of Utah has this really cool, oh shoot, I only have part of it on there. University of Utah has a really cool thing, and I'm sorry, I have it partially cut off. This is a coffee bean, and it says that over to the side, but I have it cut off, and I apologize. But anyways, you can go on this from learngenetics.utah.edu, and you can type in size and scale of cells. But anyways, as you scroll through here, you can see that here a single cell for a coffee bean or a sesame seed, they put a grain of salt, even though that's not a cell, and then a grain of rice. You can see their size, okay? And if they were blown up to this big, which this is probably the way you're viewing it is probably bigger than a coffee bean, but if they were that size and you zoom in slowly, we can get down to what's called an amoeba and a paramecium and even a human egg and skin cell and the photoreceptors in the back of your eyes and a human sperm cell. There's the human sperm cell compared to the egg. Look at the size difference. It's a huge difference. And then one human chromosome and a red blood cell and yeast, Baker's yeast. And then as you keep scrolling, you're down to E. coli. This is about where the last cell is in these pictures. And then beyond that, these become like either cell parts right now, or what we call organelles, or we're getting into like viruses, which we don't even consider living things. They have some living thing like qualities, but they're not considered living things. And look at how small. Hemoglobin, which carries the oxygen for you in your blood, and your transfer RNA and antibodies. Look at how small we're getting. Down to a single atom of carbon. Okay? So now we zoom back out. Woo! We're going to go really fast. And this is like the smallest like singular living thing we have on there. And then look how small that is. It just goes away. Now watch the human egg. Some of you guys will have good enough eyesight that you can see this human egg all the way down with your naked eye. Let's see. Some of you guys can still see that blue dot right there. Anyways. But you cannot see a sperm cell. But anyways, a eukaryotic cell is a true cell. And I'm sorry, I got small on this for some reason. I didn't mean to do that. There we go. We're going to go to eukaryotic cells. Eukaryotic cells mean true nucleus. True is U, and then nucleus. And in eukaryotic cells, we're going to stay here for a minute. We'll just stay here on this because it does make sense for a lot of it. Eukaryotic cells are what humans are made of. And there's a lot of compartments inside, and you can see that here in the picture. In a cell with compartments, the chemical reactions can occur in isolation from one another. And this makes the whole system more complex. Now, plants that have cells, this is an animal eukaryotic cell, but plants, they actually store their energy as starch, while animals will store their extra energy in the cells as glycogen. The biggest distinguishing factor of a eukaryote is the fact that it has this nucleus. So having a nucleus is what makes it different from a prokaryote. If a cell has a membrane-bound nucleus, then we do call it a eukaryote. Some eukaryotes are multicellular, and some are just going to be unicellular. So there's some very pretty, here's a plant cell, and you can see it's a lot more rigid. It has a cell wall. It has this rigid cell wall, and that's basically to support it and protect it because the animal doesn't, or the plants, animals have generally a skeleton to support them, whereas plants do not. So they have this harder cell wall around them to help them keep their shape. Okay, so inside of each cell, there is a plasma membrane that surrounds, here it is, the plasma membrane around the outside of the cell. And the plasma membrane, and here it is right here on the plant cell. So the plasma membrane is made up of this double layer. You can see that this outside layer is the same as this inside layer. And they both have these heads and they have tails. This is called a phospholipid bilayer, and inside of these are different proteins, and the proteins are channels that allow certain things, larger things, to go through. Now, some things can go straight through the phospholipid bilayer, but some have to go through these channels. So this phospholipid bilayer will basically form almost automatically because it has polar and non-polar sides. So it's like charged, it has sides that are charged and not charged. The inside is hydrophobic, meaning it repels water. And the reason why the inside has to be hydrophobic and repel the water is that if this filled up with a bunch of water, it would basically split it apart. So the inside just kind of, if water comes through here, it's like, and it pushes it through to the other side as fast as possible. It repels water. The outside is hydrophilic, meaning that it's okay around water. So again, the heads are polar, and that means like this red part is polar, and it's hydrophilic. It likes being around water so that you can keep your cells hydrated. But the inside of this layer is hydrophobic. It's nonpolar, so it'll push the water through. So, and then there's these protein layers that go through it, and then there's charged particles. Charged particles sometimes have a hard time crossing because they're also polar. All right, let's move on. Inside of the membrane is what's called the cytoplasm. The cytoplasm is a jelly-like consistency that all the organelles kind of float around in. And then you also have the cytoskeleton, which is made of microfilaments, which are like proteins that can make it harder and give it more shape. Some organisms have flagella or cilia, which is, it's not all the time, but they sometimes have flagella or cilia on them so that they can move really easily. I should have brought up a picture of this, but they have proteins extensions that basically flex off the back of them, so it makes currents, so they can either move, or they actually have like a waving motion towards their kind of mouth opening so that they can wave tinier little things inside of their mouth. So that's what the cilia and flagella are for. The nucleus has all of your DNA, and all of your genetic material is found inside of the nucleus. We're going to end up talking about this a lot later. And then inside of the endoplasmic reticulum, this is where proteins are made, but in the ribosomes, which are in the endoplasmic reticulum. So the endoplasmic reticulum has a rough area. The rough endoplasmic reticulum makes proteins, and the smooth part of that makes fats. Then we have, let's see, the Golgi. The Golgi apparatus, I think, is often forgotten about, but the Golgi actually faces, it's usually near the endoplasmic reticulum, and it will actually take the packages that come out of the endoplasmic reticulum. It receives the vesicles that come from it. It opens them up. It modifies it and usually adds like sugar to it. It'll repackage it. It actually tags it with different molecules so that it gets to the right place. And then it will, on the side that's not facing the endoplasmic reticulum, it will send those little vesicles, those newly packaged materials, to go out. And here's a cool picture of the Golgi that was actually taken with a transmission electron microscope. It's kind of cool to see that. Lysosomes, we'll talk about them a lot more. I think that they're forgotten about a lot, but lysosomes actually break down your, like, the unwanted things in your body, and it's actually can get cells that need to die, go, move, be on their way. It can actually get the cell to kind of dissolve and break down itself when it is time to do that. Okay, moving on. We're going to move to the ribosomes. Okay, So ribosomes, they make proteins, and they make proteins in the endoplasmic reticulum. Then we're going to move down to the mitochondria. Mitochondria are often called like the powerhouse of the cell, or they're called, like energy factories. They're actually really interesting because look at these. Um, this is one right here. This is a mitochondria, and it looks a lot like this a little bit, which is a chloroplast. So chloroplasts and mitochondria look a lot alike, and they also look a lot like bacteria. They also, separate from the whole rest of the cell, they have their own DNA. They go through their own replication. They have their own double membrane that surrounds them. So that unique feature. Now, a mitochondria powers an animal cell, whereas a chloroplast powers a plant cell. There's a lot of speculation with scientists that a long time ago, these guys were actually it's called the endosymbiotic theory, that something larger just came and consumed these little energy making factories, something like this in an animal and something like this in a plant, that they consumed them, but then because these guys were so efficient at making energy, instead of dissolving them as food, they decided to take them as a food generator into the cell. And that's how this endosymbiosis, this like almost a cell within the cell, came to exist. So whether that gets shown to be true more in the future or not, I'm not really sure. But anyways, it's an interesting endosymbiotic theory. So chloroplasts, they make energy for plants and algae, just like the mitochondria do for animals. And the benefit of having a cell wall around these guys have a really strong cell wall, and that cell wall is for strength and to give it shape. This plant cell walls are made of, by the way, something called cellulose. They're made of cellulose. Um, I think that might be it that we need to go over for this section. And then I want to go on to 3.4 and highlight a few things there. So in 3.4, we're back to this cell membrane. And on the cell membrane, there's a couple interesting things here. If you look closely, you can see these yellow parts here that are drawn here, and it looks different in every model. But these yellow parts here are cholesterol. Cholesterol gets a bad name in a lot of ways because it can collect inside of your blood vessels, arteries, and can make like a plaque where it's harder for your body to pump blood. However, cholesterol in small amounts are actually needed in every single cell membrane, and they almost act as like almost something to keep your temperature steady, so that this membrane doesn't get too hard and frigid when it's cold, and it doesn't get too soft and fluid like when it's warm. So the cholesterol kind of is like an antifreeze, where it keeps it from getting too cold or too hot. Also embedded here are proteins in the cell membrane. Here you can see these different protein channels, and they really act as the gatekeepers to determine what gets to come in and out of the cell. So they open and close to prevent passage or to allow passage of larger things. So some things are going to be able to come straight through here, like water. It's just going to diffuse straight through here. But some larger things, the protein channels will decide what gets to come through. And then there's carbohydrates. And there's carbohydrates on the cell membrane, and they help identify the cell membrane to other things and let almost like a flag to say this is what it is. The next thing I wanted to talk to you guys about is diffusion. So diffusion is basically things moving from an area of high concentration to low concentration. So it shows like a membrane here, but basically if there was anything to diffuse, like if somebody poured some dye in a swimming pool, you might get to see the dye at first, but then after a couple of minutes, there's a very good chance it is spread out so much that it's diffused and you can't see it anymore. Just like if there was something really smelly in one part of the house, probably by the other side of the house, you can't smell it anymore. But so diffusion is like the spreading out of substances from high concentration to low concentration. But osmosis, this is actually diffusion through a membrane. And what's interesting about this is that you can see of these particles here, there's a low concentration of, let's pretend the purple is just salt. Let's assume this is salt, okay? And the pink is water. So there's not very much salt here, and there's a lot, it's really salty over here. Well, to equalize out the percentage, water will actually diffuse from this side, forcing this to go up even against gravity because of diffusion and the membrane holding it back. I know that in some ways it seems hard to believe, but you don't have to put in energy, you don't have to force it to do this. It will just happen like this automatically. And this is one of the things that keeps us alive. When you have, say, a lot of salt and you've eaten a lot of salt, all of your cells, and you have a lot of salt going through, your blood system, your circulatory system, as you have that, it will actually dehydrate. All your cells will donate a little bit of water to equalize that until you urinate out and get that salt out of you. And the opposite can be true, that, if you have a high concentration of things in your cells, you'll feel like the need to drink and be very thirsty because your body is like sucking as much liquid out of your blood as it can to put it in the cells. This is how you can become like swollen and things like that, and what people call water weight, when it's not equalized. So there's different cells. Hypertonic is when they're dehydrated, so they're shriveled. Isotonic is happy little cells that are, these perfect little, almost like donut shape. And then hypotonic is when they get so full of liquid that they can actually burst. This is where people will talk about, how drinking too much water can actually kill you. I know that that's a controversy that comes up every few years, and that's actually true because your body will keep taking it in and taking it in and taking it into your cells until they will burst. So those types of things like that, I mean, it sounds safe, but it's just not. So that's that. And then the last one that we're going to talk about is active transport. Active transport is more complicated. It's called active because it needs energy to make it happen. But it's really important, and this is really important for the nervous system, to carry messages basically from your nerves to your muscles, and to carry messages just across your body to make everything happen. You have these areas where you have a very high concentration, like on this side of potassium and this side of sodium chloride. And so your body will actually pump different things across the membrane, and it actually takes energy called ATP to do this. And so why does your body do this? Because it wants to actually carry a message. It wants a certain message to go across. And so yes, it's a system that requires energy, but it's really important for your body to make that happen. Your body also has a few different methods in getting rid of, for instance, large particles or taking them in. It can do this via phagocytosis, meaning it goes and surrounds the particle and brings it in or out. Penocytosis, and then it can even have receptor mediated endocytosis, where it has basically it has to match with the receptor in order to take it in.